All right, good morning. Today is Wednesday, November 18th, and we are on to our final and last section of 430. Our long and windy road is almost done, where we are doing the nervous system. Again, I noticed this morning that no one apparently has spent any time looking at this over the past 2 days because no one pointed out to me that the materials for this section did not populate properly. So shame on all of you for not looking at this stuff ahead of time to prepare. But I do have the stuff available now properly for you. Yeah, mine too. Properly for you on Canvas. So everything should be available except for one thing, which we'll talk about in a second. Today, we'll start our discussion of the overview of the nervous system and start on our nervous tissue. And then we will talk about our brain anatomy today. I don't think we'll get to it. I think we'll just talk about it and then start working on that. Well, then, Joy, then you should have emailed me when you thought that they weren't available so that I could, that they hadn't populated properly because I had the stuff all there. It just didn't work. No, it's not a big deal, but I'm just saying that if you see an error, then let me know and I can fix it. We have a fair amount of material to cover and a fair amount of assignments for the next couple of weeks. As you can see, we've got a fair amount of assignments. We've got 2 unit reviews. Your first one is due Monday, as well as your Labster. And remember, you must complete all of that and get it 80% correct for full credit. Then Wednesday, your Unit 13 review is due. You have another Physio X because I know how much you guys enjoyed the first 2. And this one actually has 9 activities. So again, this is not the kind of thing that you've seen that you want to leave till the night before because obviously you won't sleep if that's the case because exactly. Again, they're not bad. They're just time consuming and they occasionally useful. We do have another review that is going to be created for correctness. And again, I purposely have not made it available for you yet. One of the things that we're going to be talking about a lot in this section, one of the things we've been talking about from the beginning of this is the relationship between structure and function. We know those 2 things go hand in hand. However, in the nervous system, one of the things that is going to be dramatically different is there is going to be a relationship between structure, function, and location. Right? If you think about it, if you've got muscle tissue up here in your arm and you have muscle tissue down here in your leg, do they do the same thing or do they do different things? Well, they move different parts of your body, but if you think about the muscle tissue itself, the muscle tissue itself contracts, the myosin heads pull on the actin heads, and they basically do the same thing, right? So essentially they're the same thing. You're right, they do different and they have different actions on our body. But all our muscle tissue everywhere contracts. Is the same thing true for our nervous tissue? Does this part of our nervous tissue do the same thing as this part of our nervous tissue? No, and not only is this area different from this one, but this one can be different from what's going on over here as well. The location matters. And so we're going to see that the location and the functions and the structures are going to go hand in hand in hand. So we have 3 things we have to put together, and that is what this review is about. This review is going to be about relating the structure of the neurons, the function of the neurons, and the location of those neurons. And so it is going to be important to understand that, but we obviously need to know a lot of information before we can start working on that. So again, I purposely have not made it available yet to give you some time to learn the material first before we do that. And so it won't be due till the 2nd of December. So we have a bit of time to work on that. All right, we have a couple more assignments after that, some fun labs that you're going to do at home, but we'll worry about those when we get to it. We still got a couple weeks to go before we do that. So 2, 4, 6, 3 weeks of lectures we need to get through before we get to that part. So let's go ahead and get started. You guys would have to tell me how the test was. I've just started grading them. I had class yesterday, so I've only graded about a handful of lab exams so far. And so like most lab exams, there is a wide range of scores that I've seen so far. Some people have done well, some people have not, but there's 1 vote for brutal. So we'll see. We'll see how the results come out. Sounds good. Well, don't close your eyes during the lecture, though. Close your eyes after lecture. All right. Excellent. Only cried once. Spectacular. That's what I like to hear. Excellent. Well, if you're not dreaming about anatomy and physiology yet, then you're doing it wrong. I'll tell you that right now. Well, if it makes you feel any better, I'm crying while I grade them. All right, let's dive into the material. That's what I want. Joed, this is college. You guys are adults. You paid to be here, so you can do whatever you need to do. Obviously, if you've got, I think I said, you said something about how you had an appointment or something. Yeah, obviously you got to do what you need to do. I mean, if you look at it, there are 13 people here. We have more than 13 people enrolled in the class. So this college, you're adults. Obviously, have you seen being here, getting the material, in person does make a difference. But if you have to leave and get it, then that's understandable. And thank you. I appreciate that. Then I appreciate it. Yes, obviously you can leave if you need to, when you need to. Excellent. All right. So let's dive in, talking first about our nervous tissue. Not surprisingly, as we've talked about, there is this relationship between our function and our structure. And so we need to talk about that as well. The function of our nervous system and our endocrine system, as we've talked about, is to maintain health and maintain our homeostasis. Right? Those are the two systems that, remember, we mentioned way back at the beginning of the class, are specialized for communication, to coordinate the activity of the structures and the organs of our body. And how is it that the nervous system does this communication? How is it that it maintains the health and homeostasis? Right, it receives information and then does what? Electrical signals, excellent. Although we can be more specific at this point. What did we call those electrical signals?
Impulse, stimulus.
Or the other one that I like that's up there is action potentials. Right, it uses action potentials, those neural action potentials that we talked about. It produces and sends those neural action potentials throughout our body and helps to maintain our health and our homeostasis by reflex and things along those lines. But that is only the tip of the iceberg of what our nervous system does for us. Our nervous system is responsible for our perception, everything we perceive from the outside world. We are inundated with a fire hose of information constantly coming into us, right? And we have a teacup of attention that we have that we can spend on that perceptions that are coming in. It influences and affects our behaviors, our movements, our memories, our emotions, so many different things that our nervous system does for us, right? In many ways, it makes us who we are. Incredibly important and significant and interesting. And again, yes, I am completely biased, but great organ system, important organ system. And of course, the study of it is the field of neurology or the field of neuroscience. Yeah, I fully admit that I'm biased. All right, let's talk about the basic functions of the nervous system. The basic functions of the nervous system, they're basically three and threefold, right? The first is to receive information from the outside world, right? Or the inside world. We receive some type of sensory information, some type of input, right? You see a bear with an axe, right? Then you have to process that information, right? Bear, bad. Axe, bad. Bear with axe, very bad. Right? So we process that information, we analyze that information, we store that information, we compare it to the information we have stored, and that is our integrative function. And then we have a motor function, some type of output that has to come up because of that, right? We realize that a bear with an axe is bad, so we have two choices, right? We have to either fight that bear with an axe or we have to outrun somebody in the class, right? And that that output goes to our effectors. And remind me again, because it's been a while since we've done this stuff. What are the things that can be effectors in our body? Excellent. Glands are definitely one of them. What else? Well, the glands would release our hormones, right? Glands release hormones. So glands would be one of the effectors. Muscles, although now, after we just finished the last part, we can be more specific. We don't have to just say muscles. We can say things like skeletal muscle. Any other type of muscle tissue that could be an effector? Smooth muscle.
Cardiac.
Anything else? And cardiac muscle, excellent. Those are the four things that can be effectors. And so we send out that motor output to those effectors and we effect some type of change. Right. I like the example of bear with an axe, but your textbook uses the example of, right, you see a glass of water, you realize that you want to drink, and you send a signal to a motor output to a skeletal muscle to lift that glass of water up. Now, again, we've done this here, but let's go ahead and use our whiteboard to do this as well. So again, when we're talking about, and we'll start with red. When we are talking about our functional divisions of the nervous system, and as we mentioned, there are three of them. They are sensory, they are our integration. Let's do this here. And they are motor. Sensory, of course, is where we receive information. And actually for this, I want to, oops, hold on, make this a little smaller. From the outside and the inside world. Integration is where we receive the information, we process information, we compare that information to what we already know, and this is where we make a decision based on that information. All right, so we receive that information, we make a decision based on that information, and once we make that decision, we send that information to our motor system, our motor division, and our motor division's responsible for sending the information to the effectors to affect some change in our body. And as we just finished talking about, we know our effectors can be skeletal muscle, hold on, effectors. Can be skeletal muscle, can be smooth muscle, cardiac muscle, and glands. Excellent. So again, no new information here. We've just organized it a little bit. And while we're at it, let's go ahead and draw a line here. That down there. There, excellent. All right. Now, the same way that there are functional divisions of the nervous system, not surprisingly, there are structural divisions of the nervous system. And there are two of those. Now, obviously, when we mention the functional divisions, notice by functional division, what we really mean is the direction it carries information. Notice our sensory carries information into the integration. And anybody know the term we use for carrying information into?
Transmit.
No, not a bad, no, true, but in this case, we want to transmit specifically going into something. Anybody know? There it is. That's the word I was looking for. Afferent pathway going into the central nervous system, into the integration center. And motor is coming out. So if it's afferent going in, what is it coming out? Efferent. Excellent. Now, we have two divisions structurally, and those are the central nervous system and the peripheral nervous system. And of course, is there going to be a relationship between the structural divisions and the functional divisions? Yeah, absolutely. And in fact, it is the central nervous system that is responsible for the integrative functions of our nervous tissue. And it is our peripheral nervous system that is responsible for carrying that information into and out of the central nervous system. So our afferent and efferent pathways are part of the peripheral nervous system, and our central nervous system is the part that is responsible for the integration. Now, of course, if this is the structural division, there must be structures that make up our structural division. So what are some of the structures of our nervous system?
Spinal cord.
Spinal cord, excellent. What else?
Brain.
There you go. I think this is the first time in the history of me teaching this class that someone actually said spinal cord before brain. That's awesome. That's interesting. Excellent. We have our brain and we have our spinal cord. Absolutely. Excellent. What are some of the other structures? Those are the two obvious ones I think that everybody gets and knows. What are some of the other structures?
Nerves.
I like that. However, let's be more specific. What is a nerve? Anybody know?
The pathway for the message, the action potential.
Excellent. Okay, so it is the pathway, but what was it made out of? Do you remember? We kind of talked about this a little bit in the muscle system. What
A bundle of axons.
Absolutely, exactly. Remember when we talked about, and I'll cheat and put the picture of it down here at the bottom. Remember, we already know what a neuron, a typical neuron looks like. It has a cell body with those dendrites, those little processes coming off of it that we talked about. And then it has a long axon that it uses to communicate. And so indeed, a nerve is a collection, a bundle of axons. And it's a bundle of axons in the peripheral nervous system. That tells us two other things. The first thing it does is if we have bundles of axons, we must also have bundles of cell bodies. It also means that we must call a bundle of axons in the central nervous system something different. Does anybody happen to know the name we use for a bundle of axons in the central nervous system?
Ganglions.
Close. That is one of the terms we're going to use, but not in this case. In this case, a bundle of axons is known as tracts. So tracts are bundles of axons in the central nervous system. Now, let's go back to nerves also for a second, because in nerves come in two flavors. Any idea what what the two different types of nerves might be?
Sympathetic, parasympathetic.
True. Okay, those are functional divisions, though. If you think about it, those are functions when we think of it that way. Structurally, you would identify nerves by where they come off of. There you go, spinal nerves and cranial nerves. Anyone know how many cranial nerves there are? 12 pairs of cranial nerves. You are going to learn everything you wanted to know and more about a cranial nerve. And of course, with a name like cranial nerve, where do you think those nerves come off of?
Brain.
Come off of the brain, absolutely. Like I said, you're going to learn everything you want to know and more about those cranial nerves. In fact, depending on how far we get in this section, because we always run long on this section, the cranial nerves could be as much as 25% of your lab exam. So we are going to talk a lot about those cranial nerves. And then, of course, those that come off the spinal cord are called spinal nerves. Anyone know how many of those we have?
12.
No, not 12. Just was curious if anyone knows. Turns out there's 31 pairs for those. You won't have to learn quite so much about those, but we will talk about those as well. So we have cranial nerves and spinal nerves that we have to talk about, making up the nerves. These make up our pathways. Excellent. Yep, there you go, 31 pairs. Excellent. And we have our tracks. So that also means that we must have bundles of cell bodies as well. Anyone know what we call a collection of cell bodies? I think someone said the term earlier.
Ganglions.
Yeah, a ganglion. A ganglion, ganglia is the plural, ganglion is the singular, is a collection of cell bodies in the peripheral nervous system. Yep, fun with vocabulary, exactly. So that must mean that there's a different term for a collection of cell bodies in the central nervous system. Anyone know what we call that? This is one you probably know, even if you don't know you know it. But we'll get to plexes in just a minute. No, in this case, it is your cortex, right? Your cortex is a large sheet-like collection of cell bodies in the central nervous system that covers most of the surface of your brain, but there are also small collections and those small bundle-like collections are known as a nucleus, with the plural being nuclei. So absolutely fun with vocabulary. When we think of the other structures of the associated with the nervous system, we of course have our sensory receptors that we perceive information from. We talked about some of those in the integumentary system when we talked about the skin. We talked about Merkel discs and Pacinian corpuscles and things along those lines. But someone also mentioned the term plexus. A plexus is an elaborate network of nerves and ganglia. And so obviously this is in the peripheral nervous system, because that's where we have the nerves, that's where we have the ganglia. And these plexi are actually able to do a fair amount of processing of information. Probably the best example of this is one we call the enteric plexus. This enteric plexus is found in your digestive system. When you had that nice big cheeseburger for breakfast, that cheeseburger hit your stomach and your stomach stretched. And as a result of the stretch of that stomach actually processed the information that was being stretched, and it started to churn up that breakfast, and it started to produce hydrochloric acid and started to break down that cheeseburger without any conscious thought from you or from the outside world. It's what we commonly refer to as the gut brain. You have this gut brain, which is that enteric plexus that is responsible for processing all of that information and making decisions locally. And so we have these plexes that are able to actually make local decisions out in the peripheral nervous system. And so notice when we talk about these functional divisions, as you can clearly see, our central nervous system includes our brain and spinal cord. And our peripheral nervous system then is the nerves, the sensory structures, and the ganglia. All right. Excellent. Questions on that? All right. That's a good start, but notice there's still a lot of space here in this organization. No, that I want red, not blue. You stay blue. And the reason for that is, let's talk about the sensory, for instance. When we talk about the sensory functional division of the nervous system, there are different subdivisions. And in fact, there are 3 subdivisions of the sensory branch of our peripheral nervous system. And again, those are going to involve where the information is coming from. For instance, we talked about we receive information from the inside and the outside world. Give me an example of information you receive from the outside world.
Temperature?
Temperature, absolutely, right? However, temperature, remember when we talked about temperature, is it a precise, can you put your finger in water and tell precisely what temperature it is? No, remember we did back in elementary school, you put one hand in cold water, one hand in warm water, and then you put both your hands into room temperature water, and one felt cold and one felt warm, right? It's because it's more of general information. Things like temperature, things like touch, those are things that can be perceived from lots of locations in the body. And so we call that somatic or somatosensory information. This somatosensory information is general sensations from the outside world. These are like the pain, these are like the tickle, these are like the inch, these are the temperature, things along those lines. But we also have things like someone mentioned sound. Where do you perceive sound from? Your ears, right? Can you perceive sound from your elbow? The top of your head? Your tongue? No. What else? Give me another example of something like that besides sound.
Vision.
Vision, absolutely. Where do you perceive vision?
Yes.
Your eyes, absolutely. Your eyes and your visual system is the most important sensory system of your body. And yes, I am a visual neuroanatomist, so I am totally biased, but each optic nerve coming out of your eyeball has over 1 million axons in them. Over 60% of your brain is involved in processing visual information. Over 70% of the information you receive from the world around you is visual information. We are visual organisms. Taste, another example, great. We have these things that are located in special locations. Now, you 100% are not supposed to do this. In fact, if you do this, I will take away participation points from you. But theoretically, when you wake up in the morning, the first thing you do is you rub your eyes, right? As you're rubbing your eyes, which A, you should not do, but B, if you were to push on them a little too vigorously, right? Or if you were to take your finger, and again, do not do this, and push on your eyeball, what would happen?
Hurts.
Okay, true. But you don't have to necessarily do it to the point of hurting, right? If you, yeah, okay, you see light. Exactly. You see light. If you were to push on your eyes or if you rub your eyes a little too vigorously, what happens is you see light. The reason you see light is when you push on your eye, you are increasing the intraocular pressure, which can damage the retina. So that's why you should not do it. But what happens is as you put that pressure in the eye, it pushes on the retina and it stimulates the retina. And the retina is a specialized structure that perceives light. So when it is stimulated, in this case, not by light, but by pressure, it sends an action potential to your brain and you see stars, you see light, right? This is an example of a special sensory information. So our special sensory information, so we have our somatosensory. Let's just do it this way. The different branches. We have the somatosensory branch, which is general sensation from the outside world. We have special sensory, which is specific information from special sensory receptors. This is things like smell, taste, vision, hearing, all those things that we just do in one location of our body. But remember, we also said that this is information from the inside as well as the outside world. Now, can you tell me, anybody here, tell me right now what their internal body temperature is? Right now, this instant. Now, yeah, you could cheat and go get a thermometer and stick it in your mouth.
37.
Okay, do you actually know that for a fact? Is it 37 precisely right now?
Probably yes.
Okay, but you said probably, you don't know for certain. How much calcium that you have in your blood? Can anyone tell me how much calcium they have in their blood right now? Sodium in their blood, what the pH of their blood is? No, we aren't consciously aware of that information, but do we need to know that information? Do we need to know how much calcium is in our blood, how much glucose is in our blood, what the temperature of our blood is, what the pH of our blood is? Absolutely. And are there parts of our body that are able to sense that information and send it to our nervous system so that we can process it? Absolutely. And so what we have with that is what we call our visceral or autonomic sensory. We have what are called interoreceptors. These interoreceptors tell us what is going on inside of our body. We're just not consciously aware of them. So yes, we have chemoreceptors and osmoreceptors measuring our blood pressure, measuring our pH, measuring how much glucose we have in our blood, measuring all of those things and processing that information, sending it to our brain so that that information can be processed and we can make decisions based on it. So 3 different types of sensory information, 3 different sensory subdivisions. And if there are subdivisions of our sensory branch of our nervous system, do you think there are subdivisions of our motor branch of our nervous system? Yeah, absolutely. For this, there is 2 subdivisions. Switch to brown, I think that looks nicer. Excellent, 2 subdivisions. And not surprisingly, those 2 subdivisions are based on the effectors. And this one I think you already know, even though you may not know you already know. What branch of the motor nervous system controls our skeletal muscle? We just finished talking about that. What kind of neuron did we say sent information to our neuromuscular junction to get our skeletal muscle to contract?
Voluntary.
Somatic. Somatic motor. So our somatic motor controls our skeletal muscle. And anyone know the name of the branch that controls our smooth muscle, our cardiac muscle, and our glands?
Autonomic.
Autonomic, excellent. Autonomic motor, which also has 2 subdivisions. And what are the two subdivisions of our autonomic nervous system?
Sympathetic and parasympathetic.
Sympathetic and parasympathetic. We will talk about our sympathetic and parasympathetic branches of our autonomic nervous system in great depth at the end of this section. But for right now, there are two little cutesy mnemonics, two little cutesy rhymes that help you to remember what our sympathetic and parasympathetic does. What is the little cutesy rhyme for the sympathetic? Tells us what the sympathetic nervous system does.
Vessel constriction of vessel constriction when like
So you're giving me an example of what it does. An example of what it does.
Increasing the blood pressure, like when the vessel constricts, so the blood pressure increases or go up. So this is the job of the sympathetic. And when there's muscle relaxation, so this is the job of the parasympathetic.
Okay, so you were giving me some examples of some of the things that they do. However, that's not a cutesy mnemonic to help us remember in general, and I think it looks like Allison got that. Remember, that bear with an axe comes in, it helps us to deal with a stressful situation. That's what our sympathetic does, right? And as we mentioned, you either have to fight that bear with an axe or you have to outrun somebody from the class. So that is our fight or flight processing. And parasympathetic is what we're going to be doing next week on Thanksgiving, and that is our rest and digest. Now, as we'll see, fight and flight is actually a really good mnemonic for the sympathetic. Rest and digest does not do the parasympathetic justice, so we'll come up with some better cutesy mnemonics. But for the start, I think fight and flight and rest and digest are things that most people have heard of, so I think that's a good starting point from there. So there you go. Here we see the organization, both structurally and functionally, of the nervous system. We've done it here on the whiteboard, but let's go through it, continue to go through it again with the pretty pictures from your textbook. So again, our nervous system is made up of billions of neurons and trillions of glial cells, or what are also known as neuroglial cells. And I will use those terms interchangeably. There are support cells, and we'll talk about their function in just a minute. And they form the structures, like we talked about, the brain, the spinal cord, the cranial nerves, the spinal nerves, the ganglia, the sensory receptors, the plexes, all those things that we talked about. They are responsible for forming all those structures and more that we did the list of on the board. And as we talked about, these structures are organized into the different branches. We have our central nervous system where we do our integration. This is that command center that we talked about in our homeostasis when we talked about, and of course, this is the brain and spinal cord. And then our peripheral nervous system, which is the lines of communication, that afferent coming in and that efferent going out. So again, afferent means going into the central nervous system, and motor means, efferent means going out of the central nervous system. So we have those two directions as to the two branches of the peripheral nervous system. But again, as we also talked about, there are subdivisions. We had our somatic sensory, our autonomic or visceral sensory, and then those special receptors, our special sensory. And then for the motor, we had our somatic motor, our autonomic motor, which had the two branches, the sympathetic and the parasympathetic. So in this fashion, we have identified and described both the structural and the functional organizations of the nervous system and how they relate to each other. All right. I've done it on the with words on the board, but your book has this pretty picture that does a nice job of showing this as well. Notice the only thing this picture is missing is we need an eyeball or an ear or something like that to show our special sensory. So if we cheat and add the picture, then we have everything that we need. Here we see our central nervous system comprised of the brain and spinal cord. This does make up an important point. Remember, our spinal cord and our brain are the central nervous system. Their job is to receive information, process that information, and make decisions based on that information. So our spinal cord does indeed make decisions. What do we typically call the decisions that are made by our spinal cord? I know you guys know the answer to this. You just probably haven't thought of it in these terms. What do we call it when your spinal cord makes a decision based on information and sends it out to an effector? A reflex, absolutely, exactly. That's what a reflex is, right? When the doctor hits you in the knee with his hammer, you don't have to consciously think, hey, right, I should kick my leg out as a response to that. Or if you put your hand on something hot, that information doesn't go to your brain and you go, hey, my hand is on fire. This is a really bad thing. I should move it. Yeah, that's a good idea. Let's do that. And then you consciously move it as a result of it, right? You put your hand on something hot and you remove that hand before you're even aware of the fact that you should be cursing, right? You curse after the fact as a result of it. And those are the reflexes. So the spinal cord absolutely is part of the central nervous system and is part of processing. All right, we have our peripheral nervous system, our afferent pathway, and our efferent. Afferent is the sensory, and three different branches of that. We have our somatic sensory coming in, general information from large portions of the body, like touch. Visceral information, which is things like what the pH in your blood is, or the stretch of your stomach, or things like that that we're not consciously aware of. And then, of course, all special sensory, taste, smell, hearing, vision, things like that. We have our motor or efferent division, including our somatic motor, which controls our skeletal muscle, our autonomic motor, which controls our smooth muscle, our cardiac muscle. Oh, and we need to put a gland in there. So let's put a little gland, little bubbly gland. There you go. And that gland in there. And as we know, there are two branches of that as well. Our sympathetic, our fight and flight, and our parasympathetic, our rest and digest. All right, and so with those two tiny additions, we've got this great picture from your textbook that shows all of both the structural and functional organization of our nervous system. All right, questions on this? All right. It is a little bit early, but this is a good natural stopping point because we want from here, we need to start talking about the tissue itself and talk about the anatomy of these things in more detail. So even though it's a little bit early, let's do this. Let's go back to my whiteboard picture so that you guys have that to look at during the break. And then what we will do is we'll go ahead and take our first break here, take a 15-minute break, come back at 9 o'clock. And at 9 o'clock, we will pick up the lecture from here. So we will restart at 9 o'clock. All right, any questions? And I will start the recording at that point too. Any questions before we take our first break? All right, I will see you guys in 15 minutes then. So let's move on to more specifics about the anatomy of the nervous system. As we've already talked about, and we've talked about our epithelial tissues, we've talked about our muscle tissues, we've talked about connective tissues, we now finally get to talk about nervous tissue. Tissues, like all tissues, nervous tissues, are made up of cells. And there are primarily two types of cells that make up our nervous tissue. Those two types of cells are our neurons and our glial cells, or like I said, what we also call our neuroglial cells. Neurons, we've already kind of talked about when we were talking about our muscular system, are the functional cells. They're the ones that are producing the electrical signals and sending the electrical signals. Some neurons can be over a meter long. You have neurons whose cell bodies are located in the small of your back, and they have to communicate with your big toe, so that when you step on that Lego walking around the house, you get that excruciating pain associated with it. Right. So that's like a meter in length. If you're someone like LeBron James, or, that's something like 45 feet from their toe to the small of their back. So these neurons can be super long, and they're highly specialized. And we'll talk a lot about how they function. And they have characteristics similar to some of the ones that we saw in our muscle tissue. Neurons are irritable. Someone remind me again what it means to be irritable. And if you're a 49er fan, you're pretty irritable right now. Okay, so it receives a stimulus. Being irritable means that it can receive a stimulus. And what happens when it receives that stimulus? It responds to it. There you go. And it responds the same way that our muscle tissue responded. Remember, we said our muscle tissue could also be described as being electrically excitable, so that when it is stimulated, it produces an electrical signal. And as we talked about, we call that electrical signal a potential. Excellent. So again, we can say it is electrically excitable, able to produce that big potential, spread it long distances if we need it to. And much like the sarcolemma of our muscle cell, the plasma membrane of our neurons have conductivity. Right? Conductivity means that it can spread an electrical signal. Right? Again, I have to work on my analogies because normally I use these all in the classroom. In the classroom, I have a big wooden yardstick. And at the end, it has those metal nubs. And so as I took that wooden yardstick, and if I were to slam it into the light socket like I usually do at this point during the lecture, we get big sparks and smoke that comes out and everything as a result of that. But am I damaged by that process? No, because while that metal bit that I stick in the light socket closes the circuit and makes all that smoke and everything, that electrical signal can't spread up the wood because the wood doesn't conduct it. If I tried to do that with a metal yardstick, it would be a different story, right? Because that metal yardstick would conduct the electrical signal and it would spread up to my hand, and what's left of my hair would stand up on end, right? So that ability to send that electrical signal from the tip of your toe all the way to the small of your back, that meter in length, is an important characteristic of it. Just the same way we talked about how an action potential spreads down the sarcolemma of our muscle cell, it spreads down our neuron as well. So our neurons do a lot, super important, super specific and specialized cells that we will talk about. And the second type of cells, those glial cells, are what we commonly think of as support cells. We know their job is to help the neuron to make those potentials and spread those potentials and maintain the neurons and protect the neurons and all the types of things like that. But notice there's a little asterisk by that, because starting about 10 years ago, our understanding of these neuroglial cells started to change dramatically. And part of that started with the exploration of an individual's brain by the name of Albert Einstein. Some of you may have heard of Albert Einstein. He was a guy that was incredibly clever, and then he died. And when he died, he left his brain to science. Now, his brain ended up going on a mystical, magical journey, but eventually, many years later, some neuroscientists got a hold of his brain, and they were all excited because they were going to be able to examine his brain and look at his brain and be able to see all of the amazing different connections that his brain made that allowed him to make these tremendous leaps in logic that us mere mortals were not able to do. And so they cut up his brain and they looked it up under the microscope, and it turned out that the arrangement of neurons in his brain are identical to the arrangement of neurons in your brain and my brain. In fact, the next thing you should do during the next break is call up grandma on the phone, tell grandma, hey, grandma, guess what? I have the exact same neural network as Albert Einstein, and she'll be really impressed, and she'll send you 20 bucks in the mail. But his brain wasn't identical to a typical brain. While his neural network was very similar to what we see in ours, there were some regions of his brain that had as many as 3 times as many neural glial cells as a typical person does. And these types of findings really shifted our understanding of what these neuroglial cells may do. And now we're studying them in a much different way. They may play a much more important role in storing memories or processing information or making leaps of logic. So should you take this class again in 10 years, and I don't have any idea why you would, but if you did, we may be talking a completely different story about these neuroglial cells at that point in time. But for now, we'll stick with the company line. We know the things that it does do as far as supporting our neurons. And so that is what we will focus on for now. But again, it is a really, really interesting area of study that's going on right now. So there's probably a lot more there than we understand. All right, let's talk about our neurons first. Here we see a typical neuron. This is something we have already drawn in this class numerous times and talked about it numerous times. So this isn't anything that is terribly exciting and different, but let's talk some more anatomy. As we know, there is a big, large, centrally located cell body. I'll use green for this. No, dark green. So again, we have this big, huge, centrally located cell body known as the soma. And like most cell bodies, this is where you find things like the nucleus. This is where you find things like the mitochondria. They got a Golgi apparatus in here. All of those types of things that you would expect to see inside of a cell body. But there was something really interesting that they found inside of this neuron. Really 2 interesting things. The first interesting thing were these things they called the Nissl bodies. What they found, I'm going to clear my original drawing. What they found when they looked at these neurons under the microscope, especially using a special stain called a Nissl stain. A Nissl stain is this kind of bluish, purplish stain. What they found is that this neuron had a tremendous amount of really dense, dark purple material inside of it. And since this showed up primarily in the Nissl stain, they called these things originally the Nissl bodies. However, as our microscopes got better, as our technology got better, what they found is that these Nissl bodies were these large, flattened plasma membranes that were interconnected. And they had all of these dark, dense dots all over them. What does that sound an awful lot like?
Rough endoplasmic reticulum?
Exactly. It turns out the Nissl bodies were rough endoplasmic reticulum. So our neuron has a massive amount of rough endoplasmic reticulum inside of its cell body. And if only someone remembered what our rough endoplasmic reticulum did. What does our rough endoplasmic reticulum do again? Made something, absolutely excellent. Makes proteins, absolutely. Makes proteins. After all, we know this neuron is going to communicate by releasing a chemical signal. What's the generic term for that chemical signal that this neuron is going to release to communicate with another cell?
RNA?
No, what was the chemical that it releases? The generic term for it. A neurotransmitter. So this cell communicates by releasing a neurotransmitter like acetylcholine that we talked about at the somatic motor neurons. And so it needs to be able to make massive amounts of these proteins that it can then be able to release to communicate with other cells. It also has to be able to transport those neurotransmitters all the way down to the synaptic end bulb. Remember, at the synaptic end bulb could be a meter away. And our cell body has all these processes coming off of it. So the other thing that there is a large number of inside of this cell are microtubules. Microtubules, the cytoskeleton, helps to give the shape and the structure to this. So the other thing that we'll see a lot of inside of these are these microtubular structures, giving it a strong cytoskeleton, and these we call the neurofilaments. So in our cell body of our neuron, we will see a large number of Nissl bodies, a large amount of rough endoplasmic reticulum, and we'll see a lot of linear neurofilaments, which is basically the cytoskeleton. These cytoskeletons, these microtubules, remember, can run the entire length of the axon. Remember way back in chapter 3, when we talked about cells, we talked about those motors that are able to basically take a vesicle and walk along the microtubule and carry it that meter-long length all the way down to the synaptic end bulbs. So while the soma is fairly typical, there are two specializations to it that we will see inside of it. We will see those neurofilaments in the Nissl bodies. The other thing that is different about our neuron is, as we talked about, our neuron has processes off the cell body. A process is a term that we learned way back when we were talking about the bones, and a process is basically just something that sticks out. And there are two types of processes that we will find on a neuron. The first are the dendrites. Dendrites are typically short. They're typically elaborately branched. And their job is to receive information. Yes, the Nissl bodies are the rough endoplasmic reticulum. That is correct. So dendrites are short, they're elaborately branched, and their job is to receive information. This is where the information comes into the neuron from these dendrites. But as someone also pointed out, the second type of process is our axon. As we know, we have just one axon. It is unbranching, and it is long. Like we said, some of them can be as long as 3 meters or 3 feet, depending on what you like. And its job is to send information. Now, if we are going to send information 3 feet distance, that means our axon must be very specialized. In particular, it has a special plasma membrane. The plasma membrane of our axon is specialized. If I remember correctly, the plasma membrane of my muscle cell was specialized, and so I gave it a special name. What was the special name of the plasma membrane of our muscle cell?
Sarcolemma.
Sarcolemma. So the special plasma membrane of our neuron, our axon, is called an axolemma. So that special plasma membrane of our Let me cheat, move this down here, move this up here so that I can erase this and cheat and do this. So our axon has this special plasma membrane. We'll talk about what makes it special in a little bit later, but it has a special plasma membrane that we call axolemma. Yes, anatomists do indeed hate you. Now, what this means is that the cell body and the dendrites do not have this same special plasma membrane to them. So what this means is that only the axon can produce action potentials. It has this special axolemma that allows it to produce action potentials. Whereas the cell body and the dendrites plasma membrane is not special and it cannot produce action potentials. But what that means is there has to be a transition area. This transition area is the area where our plasma membrane goes from being not special to being special. And this transition area is what is known as the axon hillock. In the axon hillock, our plasma membrane is slowly changing. Now, let's think about this. If this is a transition area, do you think that right here at the very beginning of our axon hillock, we're going to be able to, it's going to be special enough to produce an action potential? What do you think? At the very, very beginning of the axon hillock, by just making one tiny change, do you think that's going to be enough? No. Or what about right next to that? Do you think it's going to be special enough there? No, probably not. However, is there going to be some point within it that will be special enough? Is there eventually going to reach some region where this has become special enough where now, boom, it's going to be able to produce an action potential? Absolutely. And so this specific region, and I'll sneak it down here so that we can write it, is what we call the trigger zone. Trigger zone isn't going to be something we're going to be able to see with the naked eye. With the naked eye, we're not going to be able to tell, this is the precise area where it is changed enough to produce an action potential. But what we do know is somewhere in that axon hillock is an area where it's become special enough where we can produce our first action potential. So that trigger zone is where we finally can produce an action potential, where it has become special enough where we can produce that action potential. All right. Questions on that? So again, we have our dendrites, we have our axon, we have our axon hillock that contains our trigger zone. And that is everything that's happening up here at the proximal end of our typical neuron. The good news is we know what the end of it looks like. We know that eventually that axon splits to form our axon terminals. Again, remember, as we talked about, there are numerous terms for these. I will consistently use the terms that I've used when we were talking about skeletal muscle, but notice your book uses the terms telodendria to talk about those branches. It uses the term synaptic terminal for what we call the synaptic end bulb. But remember, I also mentioned that there are easily a half dozen terms for this structure. Terminal buttons, synaptic boutons, synaptic end bulbs, synaptic terminals. Again, you can use any appropriate anatomical term, but I will consistently use the ones we used in the previous one just for consistency. Axon branches to form the axon terminals. And then we have those specializations, the synaptic end bulb. And Remind me what's so special about the synaptic end bulb? What are the two specializations that our synaptic end bulb has that allows it to communicate with its target cell?
The neurotransmitters.
Excellent. It contains vesicles.
Yeah.
With neurotransmitter. And what was the other specialization of our synaptic end bulb?
Voltage gated calcium.
Excellent. Voltage gated calcium channels that control the release of that neurotransmitter. So there you go. So again, down here at the distal end of our neuron, we kind of already know what's going on there. We've learned this anatomy already. So nothing new down here. All right. Questions on that? Now, notice one last thing. We keep using the term typical neuron. Not all neurons, but typical neuron. That means that there are going to be some differences in our neurons. And neurons are going to be different in two ways. Guess the two ways neurons are going to differ from each other. You guys are overthinking this. Okay, well, location, true. So I guess technically three ways. Location is one of them. What would the other two be?
Shape.
Structure and
Function?
function. There you go. Exactly. So when we talk about neurons, and let's go ahead and go back to, well, actually we can cheat and go here. When we talk about neurons, let's go ahead and go to my whiteboard. When we talk about neurons, we are going to distinguish neurons structurally and functionally. So there are going to be functional classifications. And there are going to be structural classifications of neurons. Now, for the functional classifications, of neurons, there are going to be three specific types. Anybody know what those three types might be? Let's actually do this way. If we do it here. Oh, butchered this. Still butchered it. Actually, three specific types, functional classifications of neurons. Let's do it this way. Three specific types. And then we can draw our line here. That'll give me enough room. When we talk about function, we talk about basically the direction they carry information. And this one we kind of already know. One way we can carry information is into the central nervous system. And what did we call that movement towards the central nervous system? What was the directional term for that?
Afferent?
Afferent. And what type of information did we carry afferently?
Sensory information.
Sensory, excellent. So our first functional classification is sensory. Into the central nervous system. And that, of course, is afferent. Is that going to fit? Not quite. I'll cheat and do that. Perfect. So then guess what the second one is?
Efferent.
Efferent information. Which, of course, means that it is going out of the central nervous system. And what kind of information would that be?
Integration or analysis of the information.
No, if it's efferent, what is it?
So motor.
Excellent. But you are correct. The third type of neurons are what we call interneurons. Interneurons basically are between the sensory and the motor. And these are ones that are located in the central nervous system. So they are neither efferent nor afferent because they are in the central nervous system. And they carry information between the two. All right, lift this one up there, move this one down there, and that works. So there you go. Those are our functional classifications, and that is pretty straightforward. So then let's talk now about our structural classifications. Our structural classifications of neurons are based on the, I should say there are three specific types, be consistent. And this is based on the number of processes coming off of the cell body. Let's see an example of this. So let's say, for instance, we have a cell body, big centrally located cell body. And this big centrally located cell body has one dendrite that feeds into it. Now, again, this dendrite is capable of branching. Let's just do that. Where it can have branches coming off of it, but only one process that goes into the cell body. And then, of course, we know one axon coming out. And we know that axon is capable of branching to form those axon terminals to the synaptic end bulbs. So notice, again, we have one dendrite, way too big, one axon. So how many processes do we have coming off of this one?
Two.
Two. So we call this a bi, because bi means two, polar neuron. Now notice again, we know the direction it's going to carry information. It carries information, receives it in the dendrite, through the cell body and out the axon. So this is the direction it carries information. And we'll see that that is consistent for all of our neurons. So let's look at a separate type. Again, big centrally located cell body. But what's interesting about this one is this one just has the axon connected to it. Instead of a centrally located cell body, the cell body is actually off to the side to the periphery. And it just connects to the axon. Now, of course, the axon is capable of branching at the end and forming those axon terminals, forming those synaptic end bulbs. But in this case, the dendrites actually connect to the axon. They don't actually connect to the cell body. So we still have dendrites that receive the information. We have one axon that connects to the cell body on the periphery. So how many processes does this neuron have on it? One. So guess what we call this one?
Unipolar?
There you go. A unipolar neuron. Now, notice neither of these look like what we've said was a typical neuron. A typical neuron, as we've talked about, has, oops, wrong color, has a big centrally located cell body, has numerous dendrites that connect to it. Nope, again, wrong color. So we have our branching dendrites that come off of it. So we have 2 or more dendrites that connect to it. But again, we just have a single axon that comes off that can branch to form our exon terminals. So notice this one has 3 or more processes to it. So it's not a unipolar, it's not a bipolar, but it has many. So it is a, anyone know? Multipolar, excellent. Multipolar neuron. And notice this is what we've said a typical neuron looks like. So not surprisingly, this is the most common structural type. All right. So now we have identified our functional classifications. We have identified our structural classifications. Any questions on these individual bits of information?
So I have a question. So to differentiate, like on the bipolar neuron, you said there is only one dendrite or 2 dendrites?
One dendrite.
One axon.
One dendrite, one axon. 2 processes, bipolar neuron.
And the unipolar?
Has just one process coming off of it, and that one process is an axon. Now, the one thing I will mention, and let's cheat and do this small. I'll do this, draw this small right next to it. Hold on. What, from a developmental standpoint, what sometimes happens with these unipolar is that it actually has 2 processes that come off. One that grows out backwards and one that grows out forward. And then they kind of fuse together. So in your textbook, you will sometimes see the term pseudo unipolar. Pseudo, of course, means false, because what happens is the axon actually starts as 2 branches that kind of fuse together. But for our purposes, we're going to use the term pseudo unipolar and unipolar interchangeably. Oops, helps if I spell pseudo correctly. Pseudo unipolar and unipolar are identical terms. So I'm not going to worry about that distinction. Oh, no, that's not what was wrong. Pseudo, there we go. Unipolar, it still doesn't look right. I have it right there. There we go. Pseudo unipolar. But we're going to use those terms interchangeably. So we can use those terms interchangeably. But again, notice it only has one process coming off of it. And that one process is an axon. This has 2 processes coming off of it. So bipolar and unipolar. Whereas this one has 3 or more processes coming off of them. So they get their name by how many processes come off of the cell body. Okay? Did that answer your question? We'll take your silence as yes then. All right.
Yes.
Okay, perfect. So We've done functional classifications. We've done structural classifications. Any other questions on these two pieces of information independently? Now, of course, if we've learned anything in this class, is there a relationship between structure and functional classifications? Yeah, absolutely. And the same is true here as well. As it turns out, there absolutely is a relationship between these. So for instance, for our sensory neurons, most of our sensory neurons are unipolar. So our unipolar neurons make up most of our sensory neurons. Most of our sensory neurons are unipolar, and our unipolars make up most of our sensory neurons. Of course, what's the key word in that sentence? Most, because most means not all. Some of our special sensory information, remember special and sensory information is like smell, is like taste, is like hearing, is like vision. In those special sensory, and only in those special sensory, we will find bipolar neurons. So bipolar neurons are only found in our special sensory. So sensory or most are unipolar, but there are a few special that are going to be unipolar. So notice what that means is for our motor, all of our motor are going to be multipolar. And all of our interneurons are going to be multipolar. And so clearly that means that our multipolar neurons are going to be all of our motor and all interneurons. So then if our multipolar form all of our motor and all of our interneurons, how are we going to tell them apart? Someone said it earlier.
Location?
By location, exactly. All of our motor are going to be in the peripheral nervous system. And all of our interneurons are going to be in the central nervous system. Now, we know that multipolar are our most common structural type. What do you think our most common functional type is? Where do you think we have the most neurons? In our sensory system and our motor system, or as our interneurons?
Motor?
Motor, not a bad guess. But where do you think most of our neurons are going to be located? Sensory, excellent. All right, not a bad guess. But where do you think most of our neurons are located? If you think of the structures that we talked about, do you think most of our neurons are located in the nerves, in the ganglia, in the sensory structures, or in the brain and the spinal cord? Brain and spinal cord. So if they're in the brain and spinal cord, if they're in the central nervous system, then what type of neurons would those be?
Ganglia.
No. What functional classification of neurons are found in our central nervous system? Interneurons. So our interneurons are by far the most, oops, I don't want that to be, I want this to be green. Most common type. In fact, something like 90%, over 90% of our neurons are found in our brain and our spinal cord. Which again, I don't think is surprising information. Those are our two biggest structures, so it makes sense that most of the neurons were there. So if you took all of the neurons out of your brain and you put them all into, out of your body, took all the neurons out of a person's body, stuck them in a bucket, reached in and pulled a neuron out, what type of neuron would you most likely pull out?
An interneuron?
Yeah, and more specifically, a multipolar interneuron. All right. So the most common type you would bring out would be a multipolar, and it most likely would be an interneuron. Now, I've done a truly amazing job drawing this like I always have, but let's take a look at the pretty pictures from your textbook. So again, our structural classification of neurons are based on the number of processes. So here we see an example of one of those bipolar neurons where we have one single dendrite feeding into it, one single axon coming out. And again, these are found in our special sensory locations like the retina, like our inner ear, like our olfactory nerve. Unipolar, notice we have a peripheral cell body with just a single axon connected to it, with dendrites at the end. And these are what most of our sensory neurons look like. And then here we see that typical multipolar neuron, two or more dendrites, one long axon. And again, all of our motor and all of our interneurons are going to be multipolar. Functionally, we identify them by the direction they carry information. Motor, carry it efferently out of the central nervous system, and those are our multipolar. Sensory are afferent, primarily unipolar, but we also have those interneurons, so unipolar, and again, like we said, a few special bipolar. And then we have those interneurons, which again are found in the central nervous system, between the sensory and the motor, and they are multipolar. Now, does it get even more specific than that? Sure, absolutely. As I mentioned, the most common type is indeed a multipolar interneuron, but do you think there's just one type of multipolar interneuron? Of all the billions of neurons you have? No, in fact, here I have two pictures of these two. Both of these are multipolar interneurons. But you can see they're dramatically different from each other. And one of the main ways they are different from each other is not just their shape, but they're also different in their location. And they're going to be different in their function. In this case, the first one here is what is known as a Purkinje cell. Why do you think it's called Purkinje cell? Good old Bob Purkinje, exactly. Bob Purkinje was a notorious neuroanatomist. Purkinje was a very, very famous neuroanatomist who loved to work on the microscope and actually was a beautiful artist. The drawings that he would do of the tissues that he would look at were truly, truly amazing. Some of his art, it's truly art-worthy, hang on your wall type of drawings that he would do. And so he would look under these microscopes, he would draw these elaborate cells that he did. Again, beautiful, famous pictures. And then of course he would stamp his flag in it. So we have the Purkinje cells here that are found in the cerebellum of our brain. There are Purkinje fibers that are found in the heart. He put his name all over everything. But for right now, we'll just focus on these Purkinje cells. These Purkinje cells are in your cerebellum. When we look at the brain, there's a little hemisphere down here at the posterior inferior part of the brain called the cerebellum. And that is where we find ththese Purkinje cells. What's really interesting about them is notice they have this big, elaborate dendritic system that looks like a broad, flat, fan-like structure. Now, I'm sure that's just the limitations of the drawing, right? No, it turns out these really are just kind of two-dimensional fan-like. If you were to take this cell and rotate it 90 degrees, all those dendrites would all line up in a row. So it has a very distinct, unique shape to it. And these are, again, found in a specific location and then in the cerebellum, and they're going to play an important role in movements of our body. The pyramidal cells, notice, get their name because of their pyramid-shaped cell body. And notice they don't have as many dendrites, but the dendrites they have are very, very long. These long dendrites allow them to receive information from lots of different locations. And these pyramidal cells are the ones that are found in your cerebral cortex, what we commonly refer to as the gray matter. These are the ones that are doing most of the processing of your brain. So when you're processing information, making decisions, right, behavior, movement, all those types of things that you use your cerebral cortex for, it's these pyramidal cells that are doing the work. So yeah, there are dozens of different types of each of these types of cells. And so here we see two nice examples. And again, different in their shape, different in their function, and they're going to be different in their locations where we find them as well. All right, questions on that? All right, excellent. That is what we need to know about our neurons. And your book's got a nice table that talks about the different types and their functions and everything that goes along with that. What we need to do now is to talk about the neuroglial cells. Again, it's a little bit early for our next break, but I think this is another good transition point, and we got a fair amount of information to cover today. So let's go ahead and take our transition here, take our next break. It's 9:47, so let's take another 15-minute break. We'll come back at 10:02, and we will restart at that point. Start the recording at that point as well. All right, any questions on that? Any questions on neurons, their structural and functional types? All right, excellent. Let's go ahead and take our next break, and I'll see you in 15 minutes. All righty, let's go ahead and get started. We've talked about the anatomy of the neurons. Now we need to talk about the anatomy of our neuroglial cells. And there are dozens of neuroglial cells. We are just going to talk about the most common types. And not surprisingly, there are different neuroglial cells for both the central nervous system and the peripheral nervous system. So that is how we are going to distinguish them. We're going to talk about the most common examples of glial cells, what they are and what their functions are. And we will do that both for the central nervous system and the peripheral nervous system. And as I mentioned, while there are dozens of these, I'm going to hold you responsible for 6. So notice there will be 4 that we will talk about for the central nervous system, and there will be 2 that we talk about for the peripheral nervous system. And not surprisingly, there are going to be similarities in their functions. What ones do in the central nervous system to maintain and help the neurons, they're going to need to do in the peripheral nervous system to help and maintain the neurons. So it shouldn't be surprising that there will be some similarities that way. So let's talk first about our astrocytes. Astrocytes are by far the most common type of neuroglial cell that is found in the central nervous system. From the pretty picture, you can get a sense of why it gets its name. It has a centrally located cell body and many small processes that come off of it. So it has a star-like shape, astrocyte, because there's a star-like body to it. As you can see, these help to form the scaffolding of the, so this helps to form the structure, the scaffolding of the nervous tissue. So it's providing that structure, providing that integrity to them. They also play a very important role in helping to maintain the interstitial fluid. Again, as we know, we need to have a large amount of sodium, a small amount of potassium in the area. But as we saw with our muscle cell, and we saw with our synaptic end bulb, ions are constantly flowing into and out of our cells. And we're going to see neurons work in a very similar way. So when there's a lot of potassium, for instance, which comes out of the cell, it's going to need to absorb that potassium. Excess potassium. Remember also, we talked about how neurotransmitters will diffuse out of the synaptic cleft. Well, we don't want those floating around and going to another synaptic cleft someplace else. So it will metabolize neurotransmitters to, again, help to break those down so that they don't go someplace we don't want them to do. And all of those functions in and of themselves are vitally, vitally important. However, none of them are probably the most important thing that they do. If you look at our illustration here, you can see many of the processes of these astrocytes wrap around these red things. Any idea what these red things might be?
Capillaries?
Capillaries, absolutely. It says it right there. That makes it hopefully an easy answer to think. Absolutely. It wraps around the capillaries and it helps to form what we call our blood-brain barrier. Right. One thing we didn't mention about neurons, but we definitely know, is like cardiac muscle, neurons, do they have a high rate of regeneration? No, in fact, when damaged, they don't regenerate at all. So we need to be very careful about them. As we know, capillary is where materials like oxygen and nutrients and hormones can come out to influence our neurons, but it's also where waste and toxins and other materials could come out as well. So we want to limit the materials that get out of our capillary and can influence our neurons. So what the astrocyte does is it adds an extra layer of protection, an extra layer of filtration. So it helps to limit the materials that can come out of the capillary and damage the neurons. Now, there are two main important things about this. The first is it a perfect protection? Right, it is forming this protection by taking its plasma membrane and wrapping its plasma membrane around a capillary. And what are plasma membranes made up of again? What is your
phospholipids?
Phospholipids. So things that are lipid soluble, like, oh, I don't know, alcohol. Can those alcohols just easily diffuse through those plasma membranes, come out and influence our neurons? Yeah, absolutely, which is why it's not a good idea to have Jägermeister before lecture. Right? After is totally fine, but before lecture, you shouldn't have it because it has that ability to influence and damage the neurons. So it's not a perfect barrier, but it helps tremendously. But here's the other thing. Like we've talked about before, as we start to learn and understand how the body works, we want to use that information to modify that information to our advantage. And this is a perfect example of this. One neurological condition we have is Parkinson's. Anybody here familiar with Parkinson's disease? A couple of you, excellent. Parkinson's disease is a degenerative neurological disorder that is commonly associated with some very stereotypical symptoms. One of those is a resting tremor. When their hand is at rest or their body's at rest, they have a tremor that occurs with it. They also tend to have, while they're trying to make a fluid motion, they can have a stopping and starting or a failure of those motions and some characteristics like that. And it turns out those characteristics are due to a lack of dopamine in our central nervous system. Dopamine is a neurotransmitter that plays an important role in a lot of factors, but one of the things that they found is that in people who have Parkinson's, they have little to no dopamine in their central nervous system. Now, the lack of dopamine isn't what causes the Parkinson's. Parkinson's causes the lack of dopamine. But what they have found is that lack of dopamine can influence those behaviors we talked about. If your motions, if your movements are constantly starting and stopping, if you have a tremor while you're at rest, is it easy to drive a car? No, but a person who has Parkinson's might want to drive a car. And what they found is if they can get dopamine into these people's brains, while it doesn't cure Parkinson's, it can lessen the symptoms. But here's the problem. Dopamine can't get past that blood-brain barrier. So if you were to take a dopamine pill, would you be able to get it into the brain? What about a dopamine injection into an arm or even a dopamine IV? Would any of those get dopamine into your brain? No. So do you know how they used to get dopamine into the brain? They would drill a hole in your skull and they would inject it directly into your brain. Now, believe it or not, even people with really severe Parkinson's are somewhat hesitant about having holes drilled in their head. So luckily, our pharmaceutical companies were very interested in this process and they wanted to find a way, to get around this. Now, obviously, dopamine doesn't just appear out of nowhere. It's made of building blocks, right? We have these building blocks and our cells put them together and they make dopamine. Well, what they found is one of these building blocks, a building block called L-DOPA. L-DOPA is a precursor to dopamine. What that means is this is a form that is made into dopamine. L-DOPA is a precursor to dopamine that it turns out can cross the blood-brain barrier. And once it crosses the blood-brain barrier, our neurons can then convert it into dopamine. So now instead of having to drill a hole in the head, you can just take an L-dopamine pill or an dopamine injection. It will cross the blood-brain barrier, get into the brain makes dopamine, your Parkinson's symptoms are decreased, and the patient is happy, the doctor's happy, the pharmaceutical company is happy, and the stockholders of the pharmaceutical company are happy as well. Everybody lives happily ever after, all thanks to this discovery of how dopamine is able to sneak across that blood-brain barrier, where L-DOPA and where a dopamine cannot. So as we've seen many, many times, once we understand how the body works, we can use that to our advantage. We can find a way around that vitally important blood-brain barrier to help us with these Parkinson's patients. So again, provides that structural support, metabolizes neurotransmitters, regulates potassium levels, and forms that structure, that boundary, as well as that blood-brain barrier. So those astrocytes, very, very important neuroglial cells. All right, questions on that? All right, our next type of glial cell are what are known as ependymal cells. Ependymal cells are epithelial cells. Remind me again what an epithelial cell does. I know it's been a long time since we've talked about epithelial cells, but someone remind me what an epithelial tissue does. Excellent. It covers surfaces. Well, our brain has surfaces. Our brain has an outer surface. And it actually has inner surfaces as well. There are actually spaces inside of our brain tissue. These spaces happen to be called ventricles. And we'll talk about those in more depth later. But these ventricles inside the brain and the spaces outside of the brain are surrounded by something. What are they surrounded by? Your brain just isn't in the skull by itself.
Cerebral spinal fluid?
Fluid, absolutely. And you're absolutely correct. That fluid is cerebrospinal fluid, all one word. Cerebrospinal fluid, which, like on an essay question, once you write out once, you can abbreviate CSF. That cerebrospinal fluid does what for us?
Protects and lubricates.
Absolutely. Well, okay, I have to be careful with lubricate. Lubricate really tends to have more to do with movement. Are there parts of your brain moving around inside of your skull? No, probably not. But you are correct in that it provides protection. It provides cushion inside of that, so that every time you move your head, your brain isn't bouncing against the surface of the skull. But notice if we put adipose in there, right, or bubble wrap, that would do the exact same thing. So cerebrospinal fluid is constantly being produced and constantly being reabsorbed. So while it provides that cushion, while it provides that protection, it also provides, so let's say this, provides cushion and protection, but it also provides chemical signals. It provides oxygen and nutrients. It can remove wastes. It can do all of those things as well. So it is constantly being produced and constantly circulating and constantly being recycled. Now, to make this cerebrospinal fluid, basically it is filtered blood plasma. It's the stuff in the blood plasma coming out, but do we necessarily want all of the stuff in the blood plasma coming out? No, and so that's where it's filtered. And that's what these ependymal cells do. These ependymal cells are what filter and process the cerebrospinal fluid. So they play a role in making and filtering and processing the cerebrospinal fluid. So they line the surfaces of our brain, and they make and they process and they filter our cerebrospinal fluid. All right. Questions on that? All right. Our next neuroglial cell is an oligodendrocyte. Right. Again, as it always seems to be the case, we always are going to run into that one alphabet soup term that you know you're going to have to spell at some point or another on the exam. And in this case, the oligodendrocyte is that here. Notice it is similar to an astrocyte in that it has many processes. However, what it does with those processes are very different. And again, these processes are extensions of the plasma membrane. And as we know, the plasma membrane is primarily made up of what again? What's our plasma membrane made up of again? Phospholipids. Fats. Well, what these oligodendrocytes do with these processes is different from the astrocytes. What they do is they take their processes and they wrap them around many axons. Notice this one has 1, 2, 3, 4 bits or 3 axons it wraps around. It can do it around doubles. Well, let's say it this way, round parts of many axons, hundreds of times. So it's not just wrapping around at once, it is wrapping hundreds of times around a small bit of one of these axons. And when it does that, it forms a substance we call myelination. So basically what it does is it forms the myelin sheath around axons. And again, in the central nervous system. So then the question becomes why? Why would we want to take phospholipids and wrap it hundreds of times around a bit of axon? Absolutely, Haley, you've hit it on the head. One of the things that it does, obviously, is provides insulation. After all, axons are like wires carrying electrical signals, and you have plenty of wires in the walls that surround you. And if you were to look inside the walls around you, would you just find a bunch of naked copper wires all bundled together and hoping for the best? No, each individual wire needs to be isolated and insulated so that a signal doesn't jump from one from the other. So when I turn on my light switch, only my light goes on and the garbage disposal downstairs doesn't go on, or the garage door doesn't open, or any number of other things don't happen. And the same thing is true here. We want to insulate each individual axon. But as Haley pointed out, what it does that's even more important than that is that it speeds up the action potential. In fact, it can speed up the action potential up to 6 times as fast as it would be without this myelin sheath. So we have this myelination to our axons that does that. Now, again, remember, it's fats that wrap around these axons hundreds of times, and it's only on the axons. So again, you may not have thought of it in these terms, but when we talk about our nervous tissue, in our brain and spinal cord, you may have heard of the terms gray matter and white matter. Right? Anybody heard of those terms before when we talk about nervous tissue? Right? Grandma, back when you were a 4-year-old and you were playing with the fork in the light socket, grandma told you to use your gray matter, stop being an idiot. Right? Well, what she was talking about is this division of these structures. Your gray matter in your brain and spinal cord is the stuff that we talked about, like the cortex and the nuclei that are made up of the cell bodies and the dendrites. Whereas the white matter is white because of all of that fat. The white matter is made up from our myelinated axons, because almost all axons are myelinated in the central nervous system. Now, of course, what's the key word in that, or keywords plural in that sentence? Almost all. Yeah, there are some that are not myelinated, but in the central nervous system, almost every single axon is myelinated. So your white matter is the stuff that is made up of your myelinated axons. Their job is then going to be to carry information from one place to another. And your gray matter, the cortex and the nuclei, right? Whereas your white matter, I guess I would say, would be our tracts that we talked about in the central nervous system. They carry information from one location to another. Whereas our gray matter, the cortex and the nuclei, are where the cell bodies and the dendrites are located. And that's where we process our information and we make our decisions and we store our information and we do all of that. So these oligodendrocytes help to define the different regions of our brain, the gray matter and the white matter, by wrapping that phospholipid, those fats, hundreds of times around the axons, insulating them, but most importantly of all, speeding up the action potential, making them work faster. All right. Questions on that?
So MS is like the deterioration of your myelin sheath.
Yep.
Is that the deterioration of these cells?
Yes, so you are right. Multiple sclerosis, MS, is a condition where the name tells you exactly what it is. Multiple sclerosis means many cracks. Basically, what happens is your immune cells are the immune cells that normally protect us from something outside of our body incorrectly attack and destroy the myelin sheaths of these oligodendrocytes. So they start to break them down. And as they attack that, this is an autoimmune disorder where your own body is attacking your own cells. And just like if you started to strip the insulation off of a wire, that would affect the function of the wire. You could have, signals that didn't make it all the way through, or you could have shorting of the signals as it goes through. So you get all sorts of different types of degenerative effects from this. You can loss of vision or decrease in vision, decrease in sensitivity, sensory information, decrease in motor function. Absolutely, because basically as you strip off this myelin, these neurons start to fail to send those signals, and you get that degenerative neurological disorder because of that. Absolutely. Yep. So in fact, absolutely. In multiple sclerosis, it is specifically these oligodendrocytes that it attacks. It attacks these cells in the central nervous system. Yep. Great question. Any others? All right. Then let's talk about the last neuroglial cell that we need to talk about in our central nervous system. And these are the microglial cells. These microglial cells are similar to white blood cells, right? White blood cells' job is to protect us from harmful outside material, to help us from pathogens, to help us to heal when damaged. And so these microglial cells are phagocytes. They're able to go to an area and destroy dead tissue, destroy pathogens, destroy things that happen this way. One of the interesting things about these microglial cells is that they are able to migrate through the nervous tissue. So they don't just stay in one location. Their job is to move around. So if there is an injury in your brain, they will migrate to that part of the brain. And like I said, if that's been an axe blade that went in there, any kind of harmful pathogens, bacteria, viruses, it will help to destroy that. But it also helps to get rid of dead cells as well. However, as we know, when a neuron is damaged, when a neuron is destroyed, does it repair with regeneration or does it repair with fibrosis?
Fibrosis.
Fibrosis. And that's the problem. Remember, this is where basically we replace the tissue with a fibrous scar tissue. And is that scar tissue going to work the same way as that neuron does? No, absolutely not. And so typically when we have this, we talk about a traumatic brain injury, where that damage to the brain occurs. And as a result of that, there is a loss of function, because scar tissue forms there, and the scar tissue forms in a way that impairs function, and that affects our ability to function normally, right? And unfortunately, that traumatic brain injury is an inherent problem with injuries to the brain. But it turns out that as we understand this, one of the things that we have found is that these microglial cells are very aggressive. When they go to an area, not only do they destroy the pathogens, not only do they destroy the dead tissue that is there, but if a neuron is injured, it will destroy that injured neuron as well. Yes, it's possible that that injured neuron could have died on its own, but isn't it also possible that that injured neuron could have recovered? Yeah, possibly. But the microglial cell doesn't care. It's just kill them all, let God sort them out. Now, of course, how do we know these things? How do we know what these microglial cells do? Yeah, neuroscientists have studied them. Now, I will state right now for the record, I was not involved in these research projects, okay? I don't know anybody who was involved. These happened many, many years ago, and I promise you that everybody involved died horrible, horrible, painful deaths, okay? So there's no reason to get your pitchforks. There's no reason to grab your torches and go running to UC Davis, but way back in the day, Davis was one of the areas that did a lot of research on traumatic brain injuries. And one of the ways they would do this is they would study these in rats. Basically, what they would do is they would take a rat and they would put it in a pool of water. Now, a swimming pool filled with water. Rats can swim, but is swimming the primary way they get around? No. So it requires a lot of energy and they don't particularly enjoy it. So what they would do is in this pool, they would put a plexiglass stand. This plexiglass stand was under the water, so it couldn't be seen. But as that rat slowly swam around inside, sometimes accidentally he would come across it. And when he came across it, the rat would then be able to stand on that plexiglass, keep his head above water, and he was happy. So then the next day, they would bring him back and they would put him in the water. And guess what he would do?
Look for the plexiglass.
Yeah, and since the plexiglass was always in the same place, he knew right where to go. And so they would time him on how long it took him to get to the plexiglass. And they'd do that for about a week and they'd take an average. Then what they would do, because they were very, very standardized with this, they would take the rat and to the rat, they would connect to its head a standard size and length tube, plexiglass tube. They would then take a standardized weight and drop that weight through the tube. So it was always a standard weight at a standard length. So it produced a standard traumatic brain injury. They would then take that rat and put him back into the water. Like I said, all these people died horrible, horrible, painful deaths. Don't worry. They would put that rat back into the water and they would measure the time that it took it to get to the plexiglass. And guess what would happen? Was he as effective at getting to it with the traumatic brain injury, do you think?
No.
No. So it would take him a little bit longer to get there. And that increased time was an indication of that traumatic brain injury and the severity of that traumatic brain injury. All right. So they did that. Then what they did is they did this exact same thing again. But with the second time, what they would do is they would immediately inject a chemical into the brain, and that chemical would suppress the microglial function. This chemical would suppress the microglial function. So they induced the exact same traumatic brain injury, but now they stopped those microglial cells from accumulating and from aggressively destroying that tissue in that area. And what they found in those situations is that the rats had less traumatic brain injury. With that, hold on, change the tone. With that suppression of the microglial cells, it took it less time to get to the plexiglass, and the traumatic brain injury was significantly less. Now, yes, we've tortured these poor rats, and like I said, I'm sure all these people died horrible, painful, slow deaths as a result of that. But it is studies like this that has helped us to appreciate and understand how brain traumas affect us and ways that we can minimize the damage. while we don't like to think of it in terms of rats, these types of studies in rats have helped us to dramatically increase things that happen with people when they're in car accidents, or probably the most obvious example where people see it a lot is in sports. Even 10 years ago in sports, right? If you were a football player running down the field on a kickoff return and you got hit on top of the head, you had a traumatic brain injury as a result of it, or in high school sports or in college football, and you were lucky to ever walk again with that traumatic brain injury or that spinal cord injury. Now, with chemicals that can suppress the microglial, we know about cold compression, we know about all these things. Just a couple of years ago, there was a player for Buffalo who on the first game of the season injured his spinal cord during the first game, during the first kickoff, and he was back before the season ended. Our ability to understand how the nervous system repairs, how we can protect the nervous system from damage has changed tremendously because of this. And so now those types of traumatic brain injuries, those traumatic spinal cord injuries that are occurring, people are recovering much faster, much more significantly from those with much less long-term complications from them because of studies like this and studying this types of stuff in animals. So yes, it was horrible for the animals. Again, I'm not making any kind of moral decision on whether these things are good or bad, but what I will say is that we have learned a tremendous amount from these types that have been able to use to improve the overall lives of individuals and of humans. So we have definitely benefited from that. And we see that today in some of the dramatically different ways we deal with spinal cord and traumatic brain injuries to help to improve long-lasting function and to limit the damage that is done. So again, another example of once we understand how the body works, we try to manipulate it or modify it to our advantage. And that is absolutely the case with these microglial cells. All right. Questions on that? All right, excellent. Those are the 4 most common glial cells found in the central nervous system. So what we need to do is talk about the 2 most common glial cells found in the peripheral nervous system. And while these are different cells, you'll see that their functions are very much similar to what we saw in the central nervous system. Starting first with our satellite cells. Satellite cells get their name because of where they're located. Remember, we have clusters of cell bodies in the peripheral nervous system. And remember, we call those ganglia, or ganglion being the singular. So we have these clusters of cell bodies. Here's a cell body, and here's a cell body, and here's a cell body, all clustered together in this ganglia. Well, what satellite cells do is satellite cells basically wrap around the outer surface of the cell bodies in the ganglion. That's where they get their name. They're like satellites around, right, in orbit around the circumference of these cells, bodies. So I'll just draw a couple more. You get the idea. And here we see them with the pretty picture. Their job is basically they're similar to the astrocytes. Now notice they're not around a blood vessel, so this is not forming the blood-brain barrier. But all those other functions we talked about, these satellite cells do. They are going to metabolize neurotransmitters. They are going to maintain ionic concentrations. And they provide structure and support in this fashion. So they do not form the blood-brain barrier. There's no blood vessels that they're wrapped around. But they do wrap around the cell bodies, helping to maintain the function of these neurons. All right. Questions on that? Now notice also, here we have axons, and those axons, like the axons in the central nervous system, are going to need myelin. So we're going to need cells to form myelin around our axons in the peripheral nervous system as well. And here in the peripheral nervous system, that is the responsibility of our Schwann cells. So notice both the oligodendrocytes and Schwann cells form myelin. However, remember the oligodendrocytes can form myelin around portions of many axons, whereas our Schwann cell only forms one piece of myelin around one axon. So notice on this one axon that we see here, this is a Schwann cell, and this is a Schwann cell, and this one, and this one, and this one, and this one, and this one. So notice on this small axon, 2, 4, 6, 7 different Schwann cells are being used to form the myelin on this one axon. Whereas remember, an oligodendrocyte can form pieces on as many as a dozen different axons, pieces of axons. What I like about this picture is a couple things. Notice here we see how this cell is taking its phospholipid bilayer and wrapping it hundreds of times around the axons. And again, the key to this is, yes, it is forming insulation, but more importantly, it is actually speeding up the action potential. I make a point of emphasizing this because in our peripheral nervous system, we have both myelinated and unmyelinated axons. So let's emphasize that. In the peripheral nervous system, we have both myelinated and unmyelinated axons. Now, even if an axon isn't myelinated, don't we still need to insulate it so that the electrical signal doesn't jump from one axon to another? Yeah, and so notice that's what's happening over here on this picture that we see on the right. Notice on the right, we have a single Schwann cell associated with 3, 6, 9 different axons. Notice what it's doing is holding the axons apart from each other. But is it wrapping itself around any of these axons hundreds of times? No. So notice in this case, our Schwann cell can separate axons without myelinating them. And so all of these axons that we see here over in this example, let's make that a little smaller so it fits in this space. So all of these axons here would be examples of what we would call unmyelinated axons. They don't get the hundreds of wraps around it. They don't get the advantage of speed. So the axon on the left is myelinated. The axons on the right are unmyelinated. So Schwann cells help to form that structure, help to isolate the axons, but they can also wrap themselves hundreds of times around the axon to form that myelin. Notice we've got another picture here that does a nice job of showing this. So let me go ahead and clear this and get to the next picture. So again, here's that unmyelinated. Here we see that example of the hundreds of times around forming that myelin sheath. But notice also, as we saw in the previous picture, the myelin doesn't cover the entire axon. Notice there are tiny gaps in between the Schwann cells. These tiny gaps we can simply refer to as the nodes. And if you want to just use the term node, that is perfectly acceptable. However, they are also known as the nodes of Ranvier. Now, node of Ranvier is incredibly fun to say. And why, of course, do we call it the node of Ranvier?
Good old Bob.
Good old Bob Ranvier was the first one who discovered it and planted his name on it. Just because he has a French last name doesn't mean he was weird. I mean, French people are by nature weird, right? But, yep, so again, node of Ranvier, incredibly fun to say. But again, we can just refer to them as nodes. Exactly. Ranvier, Leduc, Target, right? All of those fun French names. All right. Questions on that? Excellent. And so there you go. Just that simply, we have now identified all of our neuroglial cells that we are responsible for. All right. Questions on that? We're doing good on time. Hold on. What do we have left? All right, this works. All right, that is it for the anatomy we are going to be responsible for. So what we can need to start doing now is we need to start talking about some of the function of this. To get to this is where things, again, it's going to be an interesting combination of stuff that is a little obtuse, but also stuff that we've been hinting at and talking about in a way. So a lot of the stuff that we talk about are things that we started discussing and hinted at when we were talking about our muscle action potential and that whole process of getting the muscle to contract. So some of this stuff is going to be review. Some of this is going to be review from the very beginning of the class. We talked about plasma membranes and things along those lines, but it's a fair amount of dense information. So again, I think it's best to go ahead and take one last break before we finish the rest of this material off. So let's go ahead and take our last break, and then we will come back from this. It is 10:48, so let's come back at 11:03. And at 11:03, we will cover the last of this material and then start talking anatomy. Start the recording at that time. All right, so any questions before we take our last break for the day? All right, perfect. So I will see you guys back here in 15 minutes. All right, any questions before we get started? We're done with the anatomy. I want to start talking physiology, but I want to make sure we don't have any questions on the anatomy we've covered before we move forward. All right. As we have talked about, our neurons are irritable. They are electrically excitable. And they have the potential to receive a stimulus and respond by producing an electrical signal that we call a potential. Now we keep saying action potential, but as we've already kind of hinted at, remember only the axon is capable of producing an action potential. So in reality, there are actually two different types of potentials that can be produced on a neuron. The first one, the one we have talked about the most, is that action potential. It is a positive signal. Remember, we talked about it being about a 100 millivolt change, a big, huge positive signal. And it is all or nothing. What do I mean by all or nothing?
There's no half signal. It's either a full signal or no signal at all.
Exactly. It's like the light switch on your wall. If you went and pushed the light switch on your wall, what happens is if you just barely touch it, nothing is going to happen. You push it a little bit harder, nothing happens, a little bit harder, nothing happens. But if you push it hard enough, that light switch flicks. And when the light switch flicks, your light goes on. Now, if you push that button twice as hard, does the light come on twice as bright? No. So it has two states, on or off. It's all or nothing. And that is exactly what an action potential is. It is a big, huge positive signal that is going to be all or nothing, always the same size. And it only occurs along the axons. And again, that's what most of the light switches in my house look like. But in the dining room, I have a special type of light switch, right? And that special type of light switch has a dial where I don't have to just turn the light on and off, but I can vary how bright it is. Like for instance, if my wife and I are having a dinner alone, because the kids are up in their room complaining about something or another, we have the lights on nice and bright so we can enjoy each other's company and talk and have a good time together. However, when the kids are down there with us, especially when they were much, much younger, when you have young kids with you at the dinner, you have that light turned down as much as possible so that you don't see the light and the mess that they're making all over the place when you're in there. So you have that ability to be able to vary the size of the signal. And that's what a graded potential is. Graded potentials are small. On average, they are about plus or minus about 2 millivolts. So when you compare that to an action potential, you can see that it is much, much smaller of a signal. But it's also variable. It could be 1 millivolt. It could be 5 millivolts. It could be 3.5 millivolts. And it could be negative 1 millivolt, right? Remember, our action potential is always positive. Our graded potentials can be positive or they can be negative. So I could have a negative 3 millivolt change or a positive 2.5 millivolt change. They're small, they're variable signals. They can be positive or they can be negative. And these typically just occur on the cell body and dendrites. Remember, our axon has that axolemma, that specialized plasma membrane, and we'll talk about what makes it special in just a minute, but it has that specialized plasma membrane that allows us to produce an action potential, where the dendrite and cell bodies just have a normal plasma membrane. So they can't produce an action potential. Instead, they can just produce these graded potentials. So we have two different types of potentials that can be produced in two different locations on our neuron. Now, to produce an electrical signal, and again, what we talked about when we talked about muscles, we'll talk about here as well. Remember, these electrical signals move very much like water. So when we talk about the movement of water, we talked about dropping that rock in the pool and producing a wave, and that wave spreads. Water and electricity move in a very similar fashion. So to get any kind of potential, either action or graded potential, there are two things we need. We need a starting point, and that starting point is our resting membrane potential. And even though we'll talk about it in a second, someone remind me what we said the resting membrane potential was?
Negative 70.
Negative 70 millivolts. Excellent. Again, there aren't many numbers you have to memorize in this class, but this absolutely positively is one of them. So we need that starting point. We need the inside of the cell to be negative. And we need a way to get charged particles into and out of the cell. And the easiest charged particle that we're able to move into and out of a cell is an ion. Sodium, positively charged. Potassium, positively charged. Chloride, negatively charged. If we can get these charged particles into and out of the cell, we can change that membrane potential. And so we need some type of ion channels. And those ion channels are going to allow the ions into or out of the cell. And when you have a charged particle moving, you're basically producing a current. You're producing an electrical signal. Let's talk about our starting point. As we know, a neuron, and I'll go ahead and draw one here, is polarized to begin with. And as was already mentioned, we have a resting membrane potential of negative 70 millivolts. Oh, that's not how you spell potential. There you go. Resting membrane potential of negative 70 millivolts. So that is our starting part. The inside of the cell is more negative than the outside. And as we also know, we have an unequal distribution of ions inside and outside the cell. So give me some examples of that. We just talked about 4 examples. Excellent. There is far more sodium, lots of sodium outside the cell. Outside the cell, lots of potassium inside the cell. Lots of calcium outside the cell. And they're not all cations. Where's our most common anion? Chloride. And where is that more, outside or inside? Where is there more chloride? Outside the cell or inside the cell? Come on, I know you guys know this. Well, aren't we basically big bags of salt water? So salt water, you need sodium and chloride. So outside the cell, there you go. More chloride outside the cell. So there you go. So then let's do the little letters. So more calcium outside the cell, more chloride outside the cell, more sodium outside the cell, more potassium inside the cell. There you go. So we have this unequal distribution of ions. And as we talked about, there are 2 driving forces. Those 2 driving forces are the chemical gradient. Oops. All right, that's probably too big. 2 driving forces, the chemical gradient and the electrical gradient. Or what we call the chemical force and the electrical force. Chemical force, of course, involves concentration gradient. So for our chemical gradient, we'll go ahead and use blue for this. Which way from a concentration gradient does calcium want to flow? If it's going to go from a high concentration to a low concentration, it's going to want to go in. Which way is chloride going to want to go? Which way is potassium going to want to go? Which way is sodium going to want to go? Excellent. Simple and straightforward. Electrical gradient involves the charge of the ion and the resting membrane potential. All right, so the inside of the cell is negative, and so we'll go ahead and use red for this. So calcium, being a positive ion, does it want to be inside the cell or outside the cell? It's positive attracted to negative? Absolutely, opposites attract. What about sodium? Inside or outside? Inside. Remember, we talked about how sodium really, really, really wants to come in the cell. We can use sodium to do work for us because its chemical force and electrical force are both in. Calcium also wants to really, really, really go in the cell. But remember, calcium makes cells do wonky things. So we want to be much more careful about letting calcium in. Potassium, though, its chemical force is out, but being a positive ion, which way does it want to go? It wants to go inside. And chloride, being a negative ion, which way does it want to go? Excellent. Remember, our cell's resting membrane potential is where the cell is happy. Remember, equilibrium isn't where there's no movement, it's where there's no net movement, in or out. Well, ions want to be at equilibrium as well. And for their equilibrium, again, it would be where there's no net movement. But for ions, that point of no net movement is going to be when the chemical force is equal to and opposite of the electrical force. Oops. So basically, when the force of the electrical going in and the chemical coming out are equal and opposite of each other, that is when an ion will be at equilibrium, when those two forces are equal and opposite. Otherwise, when you open a channel, these ions, so therefore, let's say it this way. So when we open a channel, ions move to reach equilibrium. And again, this isn't a foreign concept. We already know this. If we were to say, for instance, well, the yellow doesn't show up good. We'll use brown. If we were to open a sodium channel, which way does sodium move? Come on, which way does sodium move when we open a sodium channel? Into the cell or outside the cell?
In.
It's going to go in, absolutely. Oh, I want that to be brown. It's going to move in. When we open a potassium channel, which way does potassium move?
Out.
Out. When we open a calcium channel, which way does calcium move?
In.
When we open a chloride channel, which way does chloride move? Do you guys know?
In.
In. It moves in. Excellent. So ions want to reach equilibrium, and so that's how they're going to move. They're going to want to move through those channels to reach equilibrium. All right. So I need to erase this so we can go through the rest of this. So again, they move. Our cell is polarized because we have this unequal distribution of ions across the membrane. And of course, that is maintained by sodium-potassium ATPases. Right? So as we know, potassium has a way of leaking out, like through leak channels that are always open. Sodium has a way of coming in. Notice if one positive sodium comes in and one positive potassium leaves, are we going to change the membrane potential of the cell? No, but we'll lose our concentration gradient and we'll lose the ability to do work. So as we know, we use things like our sodium-potassium pump to bring 2 potassium back into the cell and kick 3 sodium out. Of course, this requires ATP to do this, right? So this is that primary active transport. And notice also an unequal distribution. It brings in 2 positive things, but it kicks out 3 negative things. So not only does this sodium-potassium pump help us to maintain the ion concentrations, or really the ion concentration gradients, let's say it that way, of sodium and potassium, but it also makes the cell more negative. which is what we want. We want it to be at negative 70 millivolts. All right, so it's going to help to make the cell more negative. In fact, if we were to block the sodium potassium ATP pumps, what would happen is the cell would get about 11 millivolts more positive. They've actually done studies where they've taken cells and blocked that pump, and suddenly the resting membrane potential goes from negative 70 millivolts to about negative 59 millivolts. So when a cell is at rest, a lot of its ATP is actually being used on these pumps just to maintain the normal conditions of the cell. All right, questions on that? So we have our starting point, but like we said, ions want to be at equilibrium. Oh, I didn't do that picture on the whiteboard, did I? So I've lost it now. I wonder if I can do, hold on, I need this. If I do undo, I wonder if I can get it to come all the way back. How far back? Ah, yay, perfect, excellent. All right, so as we mentioned, the cell wants to be at its resting membrane potential. That is where the cell is happy. But ions are like 2-year-olds. What do 2-year-olds care about?
Me.
Themselves.
Exactly. All they do is care about themselves, right? They will cut you to get a chicken nugget. They don't care. All they do is care about themselves. And that's what ions are. Ions want to be at equilibrium. And again, remember that equilibrium I mentioned is when the two driving forces, the chemical forces, are equal and opposite to each other. We give a fancy name to this and we call it the equilibrium potential. And each ion has its own equilibrium potential, that point where it will be happy, where the chemical force and the electrical force are equal and opposite. Now we could go through the math of how this is going to work, but I will just give you these values. Sodium's equilibrium potential, so the equilibrium potential of sodium is equal to positive 66 millivolts. So if a cell was at positive 66 millivolts, sodium would be happy. The cell wouldn't be happy. None of the other ions would be happy, but sodium is happy. So the goal is sodium will always move to make the cell, or to try to make the cell, positive 66 millivolts. Now, is it always going to be successful getting it to positive 66 millivolts? No, in fact, I'll tell you it's never going to be successful at getting it to positive 66 millivolts, but that doesn't mean it's not going to try. Potassium, on the other hand, its equilibrium potential is actually negative 90 millivolts. So these three numbers, the equilibrium potential of potassium, the equilibrium potential of sodium, and our resting membrane potential are numbers we absolutely positively have to know. So let's move this to a graph. If we were to draw a graph, as I mentioned, there are 4 numbers we need to know. Anytime you have a graph, of course, you need to label that graph. So this is going to be time down here on the bottom. And this over here is going to be voltage. And as I mentioned, there are 4 numbers we need to know. Our resting membrane potential. And what did we say our resting membrane potential was again?
Negative 70 millivolts.
Negative 70 millivolts. Excellent. So let's cheat. I'll move my graph a little bit this way and that way, so I have room to put this. So negative 70 millivolts. Excellent. We need our equilibrium potential. For sodium, and what did we say that was? Positive 66 millivolts. So we'll put that way up here. And our equilibrium potential of potassium. And what did we say that was again?
Negative 90.
Excellent, negative 90 millivolts. And we'll put that right there. Wait, didn't I say there was 4 numbers we needed to know? Is there another number we've learned somewhere along the way? Well, clearly there is. I'm asking the question. Ah, there you go. We'd also talked about threshold, although remind me again when I say threshold, what do I mean by threshold? What happens at threshold? Well, threshold causes a depolarize. Why do we get a positive change? What happens at threshold? What is it that actually physically happens at threshold? Threshold is the voltage where ions move in and out. Why do ions move in and out? Because what happens at threshold?
The voltage gates open.
Bingo. Threshold is where voltage gated channels open. And anybody remember what we said that particular voltage was?
10.
Yeah, well, it's a positive 10 change. So that means it needs to get to negative 60 millivolts. Right? So we need to get to negative 60 millivolts because it needs to be a positive 10 change. So we'll put that right here. And I appreciate that my scale is a little off on this graph, but you get the idea. The important thing isn't that you're not going to be graphing this on the exam anyway, but the important thing is to know these 4 numbers. So these 4 numbers are numbers we have to have memorized. Our equilibrium potential of sodium, our equilibrium potential of potassium, our resting membrane potential, and our threshold. Excellent. Let's leave this here for now and go back to the lecture. So we have those equilibrium potentials, the driving force on the ions through the channels. And as we talked about, that is determined by their chemical gradient and their electrical gradient. All right. Now, the other thing that we've already talked about a little bit, but I want to make sure we go through it again, is, again, if we are going to have ions move into and out of the cell, since they are charged particles, they require channels. And there are basically 2 types of channels. The first type of channels are what are called leak channels or non-gated channels that are open all the time. My favorite example of this is a door frame, right? I have 2 teenage girls. You may have them as well. So as you know, as a father of a teenage girl, they are not allowed to get away with anything. So as soon as they turn 13 years of age, what we did is we removed the door from their door frame. All right. That way they're not able to close the door. They get absolutely no privacy. And because girls cannot be trusted, they can be monitored 24 hours a day, right? No? Okay, maybe not. But that would be nice to do that, but I can't. But that's the point. The leak channel is basically just that hole in the wall. It can't be opened. It can't be closed. It is always open. And again, remember, equilibrium doesn't mean that there's no movement of ions. It means there's no net change. So while a cell is at rest, there are plenty of ions coming in and going out through these leak channels. However, if a leak channel can't be opened or can't be closed, can we change what goes through them? No. And if we can't change what goes through them, then we can't do any work. To do work, we have to be able to change it. And luckily, leak channels aren't the only types that we have. We also have gated channels. And the key to a gated channels is these can be opened or closed. So if we can be opened or closed, that means we can change the movement of ions. And if we change that movement, we can do work. We saw this with our muscle cell. When we opened those cation channels, a massive amount of sodium entered into our muscle cell, depolarized our muscle cell, and produced a muscle action potential. So by being able to open channels, we were able to do work. On our synaptic end bulb, we opened calcium channels. Calcium were able to come in, and that calcium caused the exocytosis. So notice when a channel can be opened and closed, we can change how we move ions, and we can change what it's doing. We can do work. And as we have already talked about in this class, so again, it's really not new information, there are three main types of gated channels. What are the types of gated channels that we have talked about in this class? Voltage gated. And again, with a voltage gated channel, what opens and closes the door? Changing the membrane potential. All right. And again, wasn't there a significant critical point we have to reach to get that voltage gated channel to open, close? Threshold. Excellent. Perfect. So we have that threshold. All right, voltage gated channel. I saw someone also mention chemically. And again, your book uses that funny word ligand gated. But remember, ligand gated just means the same thing. And what is it that opens and closes it here? Open and closed by what? Well, all of them undergo a conformational change. In one of them, it's the voltage that causes the conformational change. In this case, what causes the conformational change? The chemical binds to it by to the channel. Excellent. And while we may not have really used the term of a gated channel at a time, we did actually talk about what the third type of gated channel is. Does anybody remember or know? I'll give you a hint. It was when we were talking about the skin. When we were talking about the skin and our sensory receptors in the skin, we talked about what the third type of gated channel was. Anybody know what that third type of gated channel? There you go. It is what we call the mechanically gated channel. With a mechanically gated channel, it is opened or closed by a mechanical force. And that mechanical force changes the shape of the plasma membrane. Remember, we talked about those lamellated or Pacinian corpuscles. Remember, those are the ones that we said had kind of that onion shape to them with all those many layers of plasma membranes in there, and they provide pressure. Because what happens is when you squeeze that, that puts a physical force on the plasma membrane. And as you change the shape of the plasma membrane, you open these channels. So there are 3 types of channels, all of them things we've talked about already. And they're voltage gated, chemically gated, and mechanically gated. And I have some pretty pictures to show this. Now let's start first with the simple pretty pictures. Here's some really simple basic examples. Here's a simple basic example of a chemically gated channel. Notice at first that chemically gated channel is closed and the poor little ion can't get through there. But when a chemical binds to the channel, in this case acetylcholine, our friend acetylcholine binds to it, it changes the shape, undergoes that conformational change we talked about, changes the shape of the channel, and it opens, and now the ions can flow through. So it can be closed, it can be opened, and it's opened by a chemical binding to it, chemically gated channel. Here is our voltage gated channel. And our voltage gated channel, this picture is a little simplistic. We will talk more about it in just a second. But what happens, actually, I'm going to cheat. I'm going to draw my picture here next to it. And then what we'll do is look at the real picture from your textbook that does a good job of showing this. I'm just going to draw simple basic subunits. So here is subunit 1 and subunit 2. They're the ones that form in the plasma membrane. So plasma membrane, excellent. Now, there is, as you can see, what we call an activation gate. These activation gates are special proteins that are embedded within this. And what happens is these special proteins are typically very positively charged. What happens when the inside of the cell gets positive enough, and how do we know if it gets positive enough? Reaches threshold. When it reaches threshold, what happens is that these activation gates will swing open. And as they swing up and open, that then opens the channel. And so we reach threshold, the activation gates open, and we reach threshold. And the channel is open. And again, as we talked about, that key magical voltage we have to get to, the negative 60 millivolts, is what we call threshold. But notice this simple illustration shows that there is a second gate. It looks like a swinging door, just like the first one, but it's really not. In reality, it is made of 2 proteins. The first protein is a long linear protein, and the second protein is a big globular protein. This inactivation gate has a very technical name to it because it has one long linear protein and one big globular protein. It is referred to as the ball and chain. Again, big technical terms. What happens when this channel is open is it's typically not very stable. And so what can happen is this ball of the ball and chain will actually swing up on the chain and it will wedge itself into this channel. Now notice, if it wedges itself into the channel is still open. The activation gates are open. So even though the activation gates are open, can ions pass through this channel? with that big ball wedged in there, can ions flow through it? No. So even though the activation gates are open, no ions can flow. And since no ions can flow through, even though it's open, we call this state inactive. So notice with a voltage gated channel, let's be more specific, some voltage gated channels, because not all of them have this ball and chain, but some voltage gated channels have 3 states. Those 3 states are open, closed, and inactive. All right, so far so good? If it doesn't make sense now, say something, because otherwise it's about to get just a teeny bit worse. All right, there's one more rule we need to learn about these inactivation gates. When that ball is wedged in there, it can't just pop out by itself. So when this channel is inactive, it cannot go back to an open state. Instead, for it to open again, so once inactive, the only way to open again is to close first. What happens is when these gates swing back down, so when they swing back down when this closes, so when these swing back down when it closes, it pops the ball out. And once that ball is popped out, then the channel could be open again. So when our voltage gated channels are inactive, there is nothing you can do to get them to open again. You have to first close, and once they close, then they will be able to be opened again. And again, this will make more sense when we look at what the channel really looks like. So we'll see that in just a second. All right. Before we take a look at that pretty picture, though, let's, excuse me, let's look at our third type of channel, mechanically gated channel. Notice at rest, this channel is closed, but when you apply a force to it, mechanical force to it, or stretch it, then that opens up the channel, ions can flow, and once that force goes away, it goes back to its original shape. So here are 3 types of gated channels: chemically gated, voltage gated, and mechanically gated. All right, and here, as promised, is the pretty picture from your textbook that does a much nicer job of showing what a voltage gated channel looks like. Notice we have the proteins that make the pore, and here are those positively charged activation gates. When we reach threshold, those activation gates swing up. So notice as they swung up, that opened the channel, and the channel is now in its open state. So here it's closed, here it's opened. But there's that pesky ball and chain. And so notice that ball of the ball and chain wedges itself in here. When it's wedged in here, it is inactive. Right? The channel's open, but ions can't flow through because it is inactive. And notice the only way to get this ball out so that it can be opened again is that we must go back to a closed state. When it closes and the doors swing down, as they swing down, they pop the ball out. So these channels go from closed to open, from open to inactive, and then they must go from inactive to close again before they can be opened a second time. Once inactive, there is nothing you can do. Plug it into a light socket, hit it with a bolt of lightning. Nothing you can do can get that to open again. It has to close first. Once it closes, then it can be used a second time. All right. Questions on that? All right, excellent. So ions want to move to reach their equilibrium potentials. Again, for sodium, that's positive 66 millivolts. For potassium, it's negative 90 millivolts. And when you have a charged particle, either entering or exiting the cell, that's way so tiny. Either entering or exiting the cell, you basically are producing a current, right? You're going to change the membrane potential. And you're essentially producing a current, right? Normally, when I think of current, again, when I think of electricity, I think of the wire going to my light switch. I flip the light switch and electrons move along the wire. And those electrons move along the wire and turn on the light in my light bulb. Well, an electron is a charged particle moving along a wire. Ions are charged particles moving into or out of the cell, crossing that plasma membrane. And so when you have a charged particle, an ion, crossing the membrane, you have the movement of a charged particle. You have produced an electrical current. And of course, what is the fancy term we have for these electrical currents? We call them potentials. Graded potentials or action potentials. So both graded and action potentials are produced by charged ions moving into or out of the cell. And that is what we're going to talk about on Monday. On Monday, we are going to describe both graded and action potentials. And this time we will talk specifically about what channels are opening, which ions are flowing, and the whole entire process. From a physiological standpoint of our tissue, the action potential is the most elaborate physiological process we have in this section. So does that mean there's likely to be an essay question on it? Yeah, there's, again, 2 or 3 ways we can talk about it. So there's probably 2 or 3 possible essay questions, but I guarantee one of them will be an essay question on your exam. And maybe more. All right. Questions on that? All right, so we will save the discussion of our potentials, how we actually make those potentials and why they're important for the next lecture. What I want to do with the time that we have left is I want to talk briefly about what we're going to be doing in the anatomy. First, if you haven't printed out your anatomy list, first and foremost, shame on you. Make sure you print out those lists. Those are your study guides. If you haven't figured out those are the things you need to be successful at this point in time, then you're likely not going to be successful. However, just as we've done, as you look at this list, there is a fair amount of material. And heck, let's go ahead and look at it. Let me bring it up. Because I guess you didn't print it out because nobody bothered telling me that it wasn't available yet. So let's take a look at the things that you are going to be responsible for. There's this one. No, that's not what I wanted. All right, so under your modules, There are three main handouts we're going to need for this first part that we'll be talking about. The first is the histology. We will talk more histology at the in the next class. We'll talk about this and go through some of the histology you're going to be responsible for. So we have this that we will talk about. And notice you've got a fair amount of histology, more than you had in the last section. So again, get used to that microscope stuff. The second thing you have on here is a cranial nerve handout. As I mentioned, as much as 25% of your lab exam could be this cranial nerve. It'll really depend on how much sensory stuff we get to. So it's a lot of material. Now, I will tell you right now, we are going to go over this handout together on Monday, November 30th. So that is the current schedule right now. But it isn't a bad idea to start getting a head start, looking at this material and trying to think of this material. However, if you get overwhelmed by it or confused by it, know that it's okay. We will go over it together, but I will tell you, your textbook does an amazing job of talking about and identifying the 12 cranial nerves and providing all of that information. So it is certainly something you could do on your own, but we will do that together. So this nice handout is there. But the other handout that you have is this one. This is the gross anatomy of the nervous system. Just like we did with the bones, just like we did with the muscles, I'm giving you a big list of material that you're responsible for. And I would say that this handout and the histology handout, these two handouts together probably is going to be about 50% of your lab exam. So there's 50% is going to be this stuff. 25% is going to be your autonomic nervous system, which there's another handout for, but we're not going to get to that for a couple weeks. And then 25% is going to be some combination of cranial nerves and sensory anatomy. So we'll see how much sensory anatomy we get to. If it's none, which unfortunately sometimes happens, then it'll be all cranial nerves. All right. Now, just like the big list that I gave you for the muscles, just like the big list that I've given you for the bones, we divided them up into parts, like the bones of the arm or the bones of the skull or the muscles that move the leg and the muscles that move the trunk. So I want to divide it up into more easily digestible pieces. And we are doing that by identifying what we are referring to as cephalons. There are 5 main cephalons for our nervous system. And let's actually look at the pretty picture from your textbook now to be able to talk about this. When your nervous tissue, get that out of the way. When your nervous tissue develops, it actually starts first as a flat piece of nervous tissue. Then what happens is a tiny crease forms in that. And when that tiny crease forms, that's called the neural crest. That neural crest then rolls up to form a neural tube. As you look at this illustration from your textbook, that tube forms really, really early at about 3 weeks of development. This tube starts to form these 5 big enlargements, what are known as vesicles, or what are also known as cephalons. And again, notice these vesicles or cephalons form very early in development. These 5 enlargements to your neural tube form at 6 weeks of development. Really, really early. So why do we care? Well, the reason we care is, as it turns out, these 5 enlargements that start at 6 weeks of your neural development, as you're still just this tiny little ball of cells, become very distinct regions of your mature adult brain. Your telencephalon becomes a region of your brain known as the cerebrum. This is the largest part. And when we think of a brain and look at a brain from the outer surface, it is the part that we think of and see. It's the big part, and I'll show you a picture of it in a minute, but it's the one that looks like a big mitten, right? You've got the big place for the 4 fingers and a little bit for a thumb. That is an absolute direct drawing of what an actual brain looks like, right? And that thing that we see when we look there, that biggest and largest part is the cerebrum, and it is formed by the telecephalon. Yes, absolutely. If that neural tube doesn't form fully or doesn't close properly, that can, spina bifida is a great example of that. Absolutely. Can be caused by decreased amounts of folic acid, for instance. One of the reasons why folic acid is so important is for the development of this neural tissue. Yep, absolutely. This tiny part called the diencephalon actually stays a region that is called the diencephalon. This tiny region called the mesencephalon stays a region called the mesencephalon. Although the mesencephalon also is sometimes referred to as the midbrain. This large enlargement, the metencephalon, becomes two big enlargements of the brain, the pons and the cerebellum. And the myencephalon becomes a region of the brain that anybody who's a fan of the Waterboy knows is the medulla oblongata. Notice they've done a nice job of color coding these regions here. And then when we look in the mature brain, we see those regions. So here, the big curly cube mitten part is the telencephalon. Deep to that is the diencephalon. Then the mesencephalon are two big enlargements from the metencephalon and the myencephalon. And again, with all these M's, they can get a bit challenging, but notice they're in alphabetical order. Mes, met, my. So just remember that, and those are them in order. Here we see again, in intact brain, they've had to kind of highlight, shade this part. If we peel apart the telencephalon or cut it in half, we see this duck-shaped place here, which is the diencephalon, this region here, which is the met, the pons, and the cerebellum. These two enlargements are mesencephalon, metencephalon, and myencephalon. And there you go. We peel the cerebellum off and we see those color-coded regions again. So when you look at your list, for instance, you will see that I have given you a list of telencephalic structures or features you are responsible for. I have given you a list of diencephalic features, mesencephalic, metencephalic, myencephalic features, and then our meninges and spinal cord. So I've tried to divide the material up into more easily bite-sized pieces. Now, some of these things on your list, like for instance, a gyrus, is just a structure. So some of these things are just going to be structures. And so you just need to be able to know and identify that structure. So when I point at it on an exam, say, what is that structure? You can tell me what those structures are. However, some of these are going to be what I will refer to as regions or areas. These regions or areas are going to have a specific function. So not only do you need to be able to identify them, so if I point at them on the brain, you're going to need to know to be able to identify, that is the prefrontal cortex, but you're also going to have to tell me what the function of that prefrontal cortex is. All right. Now, again, we will go through this list together, identifying these structures, identify these regions and areas, identifying the specific functions of these regions and areas. We will do this material together, but it's not a bad idea to get a head start at some of this vocabulary and being able to have started talking about and recognizing it. All right. So that's our game plan. Next class, we will start talking potentials. Then we will do our histology, and then we will start our brain anatomy. Any questions on any of that? All right, well then that is our game plan for today. That is all I wanted to cover for today. And we finished a teeny bit early, which is always a plus. So excellent. So any other questions on anything we've covered before we get started? Before we finish. All right, then have a good day. Have a safe weekend. Study hard. You're limited on how many weekends you have before the next exam. There's only 3 weeks left, so start looking ahead of time. You got a fair amount of homework too. That'll keep you busy. But start looking at this material, start thinking about this material, and we will get into more depth as we go forward. All right, have a good day, have a safe weekend, and I will see you guys on Monday.